Hi, this is Team Brainiacs with Ben, myself, Mary, and Stacy, and we're gonna give you a little update on some things, issues we've run into and the progress we've made. So some of our pitfalls is we had an issue with our masking in NumPy versus MATLAB, and so I had to go on a deep dive to uncover the problem. And we had problems with pickling different size data where we couldn't open the data once we uploaded it to the AWS. And now that is resolved as well. And we found that deep light requires transforming our data into a different shape. And so we didn't have the processing time to do this. But Ben is working on the deep learning portion, building a CNN for our data. So I just wanted to kind of show the masking pitfalls. So you can see in the top panel or the top picture on the right side of the brain, especially in the second Z equals -12 and Z equals 0 slice, you can see that part of the brain is still exposed without the proper voxels being covered up. And then in the bottom one, we were able to fix it. And I actually had to take all the variables and put them in MATLAB and play around with it and then look at NumPy's documentation. And so we found there's an order parameter and we used Fourier transformation and we are now winning.
Yeah.
So to recap, we have been working on single subject pipelines, looking at the best normalizations and grid search, working on group level pipelines, and then exploring deep light framework for the pre-trained model and fMRI. Scratch that one, we're actually building our own CNN. So I'm going to talk a little bit about the progress for single subject because that's been the pipeline I've been working on. So far, all data exploration components are done. I explored different normalization strategies, did a research CV for once I found the best normalization, and I started creating visualizations for the exploration. I have run all single subject SVMs with the best parameters. I've used whole brain masks, which masks out voxels not considered in the brain. SVMs will masking out regions of interest. So this is whole brain mask minus the ROIs, and SVMs on masking out whole brain except the ROIs. So the reason we're doing this is we're trying to see what areas may be driving predictions and the brain states that we're looking at in real time. So I've also been working on a schematic for the real-time feedback test that the participants see in the scanner. There are 4 runs. We have a feedback off run, which is the first run, followed by a feedback on, which is the first run where they see real-time feedback of their increased and decreased states in the nucleus accumbens. And then that's followed by another feedback on and a feedback followed by a feedback off. So in picking the SVMs on what to train, I decided to go with to train on run 2 in the first feedback and then validate on run 3, which is the second feedback on, and then test on run 4. So these are some of my results for the single subject normalization outcomes. And as you can see, for the full group, across all 52 subjects, the average was best for Z normalization. And then I kind of wanted to break it down by adolescents and young adults. And so again, you can see that the accuracies were best for the Z normalization. And then I wanted to look at what was happening in the brain when we did the normalization. So the top panel, 2 panels are % signal change for adolescents and young adults. And you can see that there's not a lot of activation or deactivation happening in the brain. And then the second 2 are Z normalization. And then the last are no normalization. And as you can see, the Z norm is actually showing us the best increase and decrease activation separation. And then I wanted to do some thresholding. So we're looking at the top 10% voxels in both activation and deactivation, which correspond to increase and decrease trials. And so again, you can see in this one that the Z normalization is the best. And then finally, I did a 5-fold cross-validation with grid search and to see which parameters were the best based on the Z normalization that I gotten in the previous slides. And as you can see, the best kernel is RBF with a C parameter of 1. And so my next steps for single subject SVM is to finalize exploration visualizations and my exploration visualization pipeline and start analyzing the final SVMs and then to visualize those findings. And now I'm going to turn it over to Stacy.
Hi. So I've been in control of looking at our data in the respect of grouping the subjects. So we have some subjects who are considered adolescent, which is, I believe, up to the age of 19, between 16 and 19 years old, and anyone older is considered young adult. And so the data here is a bit, runs a bit longer. So the things that I've been working on have been finding the best parameters for the SVM with both groups. I ran a grid search. I don't have any results to show you because when we had some pickling trouble, those results didn't get saved. So I'll have to run it again. And it was quite a long time for adolescent group, which is a little bit larger of a group. So I'll do that in the background if it matters to have those metrics available to show. So I did finish running all models for all the brain masks, which the masks are that the whole brain mask captures the whole brain and no parts outside of the brain. And then the other masks will take out parts of the brain to areas that we think might be involved in this reward circuitry. And also there are regions of interest. So those regions that we took out in the masks, we're actually just looking only at those regions. And I ran and finished running all those models for both young adult and adolescent groups. And I just started kind of playing around with some sort of metrics. And you can see here a confusion matrix on the whole brain and adolescent. Oh, that was supposed to show young adult and adolescent. Maybe I mislabeled that one. And then we have a rock curve here and some area under the curve scores. And my plan here is also just to break this up into young adult and adolescents and kind of show the different curves between the different masks and then the different regions and see if there's any interesting thing there to note. And then the next chart is, this is looking at the decision. Since we can't visualize the decision boundary easily with such high dimensional data, well, we could probably if we chose a couple of voxels, but just to get an overlook at all of our how it's predicted and the decisions that it's made, you can see that, so on the left of the yellow is the downregulation, that's the label 0. And the label 1 is upregulating, and anything that's orange has been misclassified. Can you go into the next slide? Yeah. And the next step. So what I need to finish doing here is finish all kind of the visualizations, metrics there. I have a lot of brain images also that I need to clean up, and those other the metric visuals need a little bit more cleaning. Maybe perform some further analyses if it makes sense. Also start the write-up and add to the landing page. And I've also ran some normalization histograms for our data as well. So maybe clean that up a little bit so we can add it to the report.
All right, Ben.
Cool. Yeah, all right. So we also decided that we're going to just try and also train just a convolutional neural network to see if we can get better accuracies on individuals. And the way we're hoping to do that is to train on, there's 2 runs when the person is in the scanner where they're getting real-time feedback of their ability to up and down regulate. So we're going to take those 2 runs for all of the people, and yeah, create a neural network there. And we've got the data pipeline is complete. So and the data loader is good to go. So now it's just sort of model building and parameter tuning and that sort of thing. And so we're hoping to train a whole model on all of the subjects that we have, like all 50 or so subjects, and then take a single subject and see if we can do just a little bit of training on the model that we build off of all of the images to better predict just a single subject. And we're hoping to, yeah, see if we can get comparable accuracies to SVM using a convolutional neural network. This is a little bit different than some of the most recent research that is talking about transfer learning. And so depending on, you know, how much we can get done, there might be a possibility of transfer learning too. But for the moment, if you go to the next slide, we're just, or I'm just, yeah, so I've been working on just finishing up the pre-processing. I'm gonna also dump it in a pickle file into AWS. And then, yeah, just need to work on model tuning. And then, yeah, Stacy helped with some of the parameters here, and we're thinking of doing similar to a paper we were reading about that's 4 layers with batch normalization in each one using ReLU as the loss function. And then, of course, it's kind of tough to visualize what CNNs are doing or neural networks are doing. So we're hoping to use, or I'm hoping to use this, this software called Lime, but I'm not really sure if it's compatible with 3D imaging. So we'll see. We might have to, or I might have to use some sort of like reverse engineering or something like that, depending on what we can do. But we'll see. Fingers crossed. We're hoping to get some good prediction accuracies out of this once we train the neural net. So, yep, that's it for me.
All right, so we've kind of made some general decisions. We decided on going with writing a kind of a paper blog because of the intensity of this project. And we're gonna create a landing page for the interactive visuals and charts. And Stacy, do you want to show the interaction? I'll have to stop my share.
Oh, right.
Or do you want me to do it? I have one open.
You can do it. Go on the next slide. Just the next slide should have, you just click it. And then okay, so the landing page is very much bare bones, and it's just like, it's Notion. If you've ever heard of Notion, it's kind of really cool. Yeah, so this is a small visual, but I think we can add like a link here that looks nicer to click it. So click that S3, the Amazon, yeah.
This one?
Yeah, and then it can make it a lot larger. And this is just an example of, so these are areas of activation. There's the areas of deactivation, upregulating and downregulating. So upregulating where the area of the brain is getting a lot of blood volume and oxygen exchange. And I think the red areas are where it's starting to decrease in that sort of regulation. So some areas you see here lit up, they're the big yellow spots. There's it's showing that, I think you can show like the occipital lobe in the back there. I'm not able to.
Oh, we have to go back.
Yeah, right there, maybe. Yeah, that area where Mary is the occipital lobe. So that's where your visual cortex, where you, so what Mary was saying is that, you know, they're looking at this screen in the scanner. So you're gonna see that these areas are lit up.
All right.
Yeah.
So hopefully we can add to this. I'd like to add some annotations to show what areas in the brain we are actually looking at. But that's a work in progress.
Yeah.
All right. Thanks.